All right, thank you guys for having me here today. I'm going to give a bit of background on biological control of coffee berry borer in Hawaii, and then talk more about a parasitoid wasp that we've imported to Hawaii now, and are almost ready to release finally. And I'll explain how that process has gone. A little bit of background first. We're doing this work in collaboration with colleagues over at ARS at PBOC. Melissa and Peter, and we have access to different quarantine facilities among us. So hopefully this allows us to get things moving along. You all know, since CBB arrived here since we detected it in 2010, we've been doing a number of things, including trapping. We'd use that for monitoring. Some people would try suppression. We've looked at sampling, how to do that optimally, efficiently. And other things that I'm sure you've all heard about. The biocontrol options that we have very limited in the beginning, as with the pesticide options. Biocontrol options still, the only one that we have that is reasonably effective is the use of Bavaria bassiana, Botanigard. It's the one that you can use out in the field. It's a commercially available insect pathogenic fungus. Then we have some predatory beetles that are out there and may have some impact. And then parasitoids, parasitic insects that kill the beetle. Right? In the beginning, we were interested to see if there were any that were resident here that lived in Hawaii that we could somehow magnify their impact in coffee plantations in suppressing coffee berry borer. And I'll talk quickly about that in a bit. And then we also looked at the potential to import natural enemies. And there are a number of natural enemies that are used in South and Central America or from Africa. And I'll get to that at the end of the talk. Just quickly, a quick word about the use of repellents. And this has also been plodding along slowly. We have done quite a bit of work on this repellent called verbenone, which is an anti-aggregation pheromone in bark beetles in general. It's released by female beetles on trees when bark beetles attack them to deter more beetles from actually infesting the tree and reducing competition. So we've been testing it over a very long period, and we found that we can reduce infestation of CBB by about 50% overall using this product. We can shut down traps with it, so you can see. Sorry, the lab watchdogs are here making a noise. If we put verbenone onto a trap that's baited for CBB, we can cut down the number of beetles trapped there dramatically, and it also reduces damage out in the field. So we're looking at a special local needs label right now. We're doing this in collaboration with colleagues at US Forest Service. They're looking at using this product for managing the vectors of rapid hair death, and then colleagues at PBOC as well are interested in using this in macadamia nut. So hopefully we'll have that special local needs label out soon, and this will be available for use in Hawaii. All right, so talking about looking for resident parasitoids, what I'm talking about here are parasitic insects that occur in Hawaii that attack other bark beetles. I'm sorry, I'm just going to tell my dog to shut up. I'll be right back. Sorry. And to do this, we went around and collected little beetles, native ones, and other invasive species from a whole bunch of different habitats, and we reared parasitoids out of them. If we found any, well, when we found them, we were also interested to see if they'd use coffee berry borer as a host, and so we'd expose CBB larvae or adults to the parasitoids that we were finding here. And then if we found any that would actually utilize them, we'd try and condition them. So you can actually make parasitoids switch their preference slightly. So we would try and see if we could condition anything resident here to use coffee berry borer, Hypothalamus hampii, preferentially. That didn't really work out very well. We found A few native species, not necessarily native, a few resident species, all new species that we described and still describing now. One that was particularly interesting was this one called Phymasticus, now hollow. So it's in the same genus as Phymasticus coffea, the African species, which we're looking at importing and releasing now for CBB biocontrol here. So we found this attacking other Xyloborus species, which are borers that attack various trees. We found it attacking Uwalacea fornicardus, which you might know of as Polyphagous shottle borer, which is quite interesting and potentially useful for protecting other tree species. The new species, Phymasticus hollow, does not attack coffee berry borer, so unfortunately it doesn't have potential for suppressing the pest here. We found a number of other species, and I'm just going to talk really briefly about them. In different families attacking these. They all attack, apparently, other introduced bark borer beetles here. None of them preferentially attack coffee berry borer. We got the one Cephalonomia to actually utilize coffee berry borer if we exposed them in petri dishes, and then we tried conditioning them and didn't have particularly good results. So I don't think there's very much potential in anything that's resident here, unfortunately. So that brings us to the importation approach. In other words, bringing parasitoids from the place of origin of the pest and checking to see that they're safe to release here and then releasing them into the environment. And this is a long and quite tortuous process. The permitting is complicated and it really takes a long time. I'll talk a bit more about it as we go. The particular species we're looking at is called Phymasticus coffea. It's originally from East Africa, the ones that were brought into quarantine by Seneca Fay in Colombia, originally from Kenya. And this species has been introduced into multiple Central American countries. They've had reasonable success. It typically doesn't establish there. In other words, you have to keep releasing it. You have to make augmentative releases, probably because of their ability to clear pick coffee every season. So there's no reservoir of CBB for the wasps to survive on. In Kenya, CBB is not a major problem at all. And samples taken from there suggest that Phymasticus coffea is the most prominent parasitoid of the beetles. So it gives us some confidence that it could be effective here. We do have reservoirs of CBB after picking. There are still beetles on the ground, beetles on some trees, unmanaged coffee, and so on. So we're hoping that it actually has the potential to establish. But otherwise, we will look at augmentative control. It's an interesting parasitoid. It's one of the very few that actually attacks the adult beetle, while the adult coffee berry borer is boring into the fruit, into the coffee cherry bean, and it's exposed. Then the wasp will come along and insert a stinger of ovipositor in between the rear wings of the beetle and lays 2 eggs into each beetle. And then once the eggs are inside, the larva of the wasp comes out and it consumes the body of the beetle and kills it. It's got a curiously long life cycle. It takes 48 days, depending probably on temperature, to go from egg to adult. And unfortunately, the adults do not live particularly long. They're probably live less than a week. So that's a bit of a worry, but we're hoping that we can get enough of them out there and have the population, the emergence is sort of asynchronous, so that we constantly have adult wasps out in the environment eventually. Previous work has shown in semi-field conditions with caged trees and so on in Colombia that they can get up to, I think, 75 to 90% parasitism, which is quite encouraging. If we get even half of that here, it would be very useful. Anyway, we went through the process of getting all the permits and brought this wasp into quarantine in Volcano, the Forest Service facility over there. And we got them from Colombia, where Seneca Fay have a mass rearing facility. They mass rear this insect to release into the environment there in an augmentative manner. So we could get shipments sent over to us periodically into quarantine. Here's a little illustration showing how it's sitting on the back of a female coffee berry borer. She's burrowing into the fruit here, and you can see the wasp sitting here. She has her abdomen pointed down, and her ovipositor or stinger is inserted between the back wings over there, and then the eggs are laid inside, and the larva develops as shown in this in this illustration down here. And then typically you'll get two wasps emerging from the adult beetle, one male and one female, a bit of variation. And I think each wasp can lay 30 to 50 some eggs, maybe. Probably with an average of around 38 each. So they can potentially each parasitize probably 20 to 30 CBB females. So the first thing we had to do was conduct non-target screening. That's a significant concern in Hawaii, is that any biocontrol agent that you're bringing in is safe to release, and that it doesn't attack native species, and it does not attack other beneficial species like other biocontrol agents, for example. So we had to, over a period of about 2 years, I think, we collected native scarlatine species, native bark beetles that occur in a whole range of different native plants, and often really hard to access areas, and bring those into quarantine and expose them to the parasitoids to see if the parasitoids will attack them. We also got a whole bunch of other invasive species that are present here, other invasive bark beetles, and a couple of other beetles to see whether they would attack anything distantly related to coffee berry borer, Hypothenemus hampii. Right, which I'll talk about now when you get to this illustration down here. This the results. We did these in petri dishes. They were no choice tests, so the wasps were put in. They were given one host species that they could potentially parasitize. It's an extremely conservative test. It's not so. Choice tests are interesting, but they're kind of difficult to interpret sometimes. A no-choice test is a kind of an acid test. If the parasitoid cannot use the single host that it's offered, and it just dies without laying eggs, then you are well justified in suggesting that it's not a host. Okay, so what did we find in the course of this work is that of the 7 native species that we exposed them to that didn't parasitize any, the native species are quite distantly related to the genus Hypothenemus. Hypothenemus being the genus in which coffee berry borer and a bunch of other invasive species occur. Outside of the genus Hypothenemus, we tested 27 other species. They didn't parasitize any of those either. Some of those were also introduced, or they were all introduced species, and some quite distantly related things like ladybugs and so on, just to prove that they're not going to attack beneficials. If you look at the results over here that we're showing, this graph shows the different species: Hypothenemus hampii, Hypothenemus obscurus, which is tropical nut borer, which is a pest on macadamia nut, and then a number of others, Seriatus, Burmanus, etc. And below that, you see this evolutionary tree. It shows the evolutionary relationship between these different species. And this continues way down into a big tree of all the beetles. The nearest common ancestor on the tree to Hypothenemus, I think, was 75 million years ago. So they're remarkably distant from those. And Hypothenemus hampii, coffee berry borer, is quite significantly different from all the rest. It only utilizes one host. While it's boring into its live host, the host is releasing chemicals that attract the parasitoids. However, in petri dishes, we got some parasitism and emergence of a number of these species. The white bars are the percent parasitism of the non-target, or the target, in this case, the control. And then the dark bars are the percent emergence subsequent to them being parasitized. You can see for coffee berry borer, Hypothenemus hampii, we got 75 to 80% parasitism, and about 70 to about 75% emergence. So an effective host. Obscurus and Seriatus, the next two most closely related, we got significantly less parasitism, maybe 55-60% parasitism, and then maybe 40-45% emergence. And so it reduces as the species become less and less related to coffee berry borer. And this means that they're physiologically suitable as hosts, right? The insect can utilize them as hosts to varying degrees. The less closely related they are, the less effective. In fact, the one, Hypothenemus eruditus, there was no parasitism, no emergence, of course, right? The furthermost relative. This gives us confidence for a number of reasons. It shows us that the parasitoid we're looking at will only attack Hypothenemus, right? Only attack species in that genus, and they're all invasive here. Doesn't attack any native species or any beneficials. And it confirms our theory that things that are less closely related will be parasitized, as you can see down here with eruditus not being parasitized. There's some chance when you get this out there that it might attack tropical nut borer, for example, right? And we should see what happens. Right, so we were confident that this was good supporting evidence. We published this information back in 2021, I think, and then proceeded with the permit applications, which are a complex process. You have to go through the Hawaii Department of Agriculture, Hawaii Board of Ag, and you have to get APHIS permits from the federal regulator organization. Both of them are quite tortuous processes, which I'll talk about really briefly, just to give people some perspective on that. Back in 2022, sorry, 23. We submitted, no, it must have been 2022. We submitted the draft, what we call environmental impact assessment to APHIS, the federal regulators, and to the state of Hawaii in 2023. State of Hawaii requires a cultural impact assessment as well, which we submitted along with that, and it was supportive of release of the biocontrol agents. We went through this whole process, which requires multiple reviews that get sent out to multiple interested parties. And we eventually are able to issue what we call a FONSI, a finding of no significant impact. It should be no significant negative impact, really, for both state and federal petitions. And then eventually in April 2023, we got a positive vote from the Board of Agriculture to import and release Hypothenemus coffea. And so it worked out well. It took a bit longer than we'd hoped, but it was quicker than what many biocontrol agent permitting processes are. And then subsequent to that, things have gone a little slower than we hoped. So what you have to do once you get this permit is bring the biocontrol agents into the state, into quarantine, which is technically not in the state. Okay, you have to bring it into quarantine. You have to then process it through 2 generations in quarantine, so you can prove that there are no pathogens, there are no hyperparasites, in other words, parasites of your parasite that you want to release, that there's no contamination with other things, and that you in fact do have the correct species. So you rear them through 2 generations, you check that nothing unwanted comes out, and then you confirm the identity of the things that you rear, that you're producing multiple generations of now, and you subject those to Hawaii Department of Ag. Once you do that, you're allowed to remove them from quarantine and mass rear them in your own facility. So our colleagues at ARS, at PBOC, did this. They started last year, and it went very slowly because of conditions in the quarantine at Volcano, but they were eventually able to get wasps out of quarantine. They showed that they were clear, the correct species and so on, and then I have them at PBOC up in Hilo. And I think we can double check with Melissa again, but they're looking at making releases on Hawaii Island early this year, hopefully. We've also now started our own colonies. We were able to bring, there was a permitting issue at first, allowing us only to go through Volcano, and eventually we got a permit to bring them to Oahu to raise them in the Hawaii Department of Agriculture quarantine. And this is all just because of permit conditions. If the permit says that it cannot be released from your particular quarantine into the environment, then you're stuck. It doesn't matter what you do. It's like impossible to move ahead. So we had to get this new permit to actually do this. Our first permit said that they all had to go to a particular location. Anyway, details of that don't matter. So we received was from Colombia, from Cafe in December in 2024, and we had a remarkably good emergence. They came billowing out of the beetles that we got from Cafe, and then we had them parasitized beetles in quarantine, and that looked as if they did an excellent job. We've currently got that first generation in quarantine still. We're waiting for the next to emerge, and then we'll be able to make all the confirmations that that is what we expect and that there's no contamination. And that's going very well. I'm sort of optimistic that we'll be able to get them out into quarantine by the end of this month, I hope. And then if that all goes as planned, then we should be able to make releases maybe March, but more likely April, carrying on then, right? We'd be able to make releases as we go. The plan is we get to release onto Oahu, Maui, Kauai. ARS will be supplying Big Island. And depending how rearing goes, we can do both. We can each do both. So that's the optimistic situation right now, right? We do have these things in quarantine, and it seems to be going well. Okay. Here's a quick video showing. Sorry, I've got a little question floating right here. This video shows, and these are CBB, the beetles over here, and you all know how small those are. And let me just get the video going. You can see over here where my cursor is, there's, if it was still, a parasitoid, a female, parasitizing a beetle. There's the little male. You can see how tiny he is. And another male. So you can get an idea how tiny these wasps are. And a little bit of an idea of their behavior, right? How they get hold of the beetle. And this is outside of a berry, so it's a little different from what you typically see. But they get hold of them, they grab on pretty tight, and then they start to lay their eggs inside. This is what we see in our quarantine beetles. All right. Oops. Okay, that's all I have. I'm happy to answer questions about that. Thanks to everyone for assistance with this, the funders and everyone who helped out with permits and all those things. So yeah, I'm happy to answer questions about that.